हाँ ओके ठीक है, ओके यू आर एबल टू, हाँ ऑडियो ऑन है मेरा। वीडियो में स्टार्ट करता हूँ। आप नहीं मुझे दिख रहे।
दिख रहा है?
Yes. Glad to see you, Professor, after a long time.
After a long time. Is it whether everything working or not? Sorry.
हाँ मैं no issues एक्चुअली ना मैं लेट आ रहा हूँ ऑफिस से ना, उसकी वजह से मैं भी टाइम नहीं दे पाया इतना। आई एम सॉरी फॉर दैट।
Hey, no problem. Slide just share कीजिए, click share button. Just we check is it working or not. Slide desktop पे है ना?
हाँ डेस्कटॉप पे है।
हाँ आपने Zoom पे वो ग्रीन कलर की शेयर स्क्रीन है ना?
ओके
उसको क्लिक कीजिए।
एक मिनट मैं करता हूँ। हाँ आ गया। ठीक है, हो गया ना? तो अभी हाँ मुझे सुजीत एक चीज बताइए, आपको अभी मेरा ये पीपीटी दिख रही है, उसके नोट्स भी दिख रहे हैं, मेरा ये अभी प्रेजेंटेशन मोड में दिख रहा है आपको? अभी मेरे को एक ही इशू आ रहा है मेरे ना, मैं जो वर्जन यूज कर रहा हूँ उसमें मैं मेरे नोट्स नहीं देख पा रहा हूँ लैपटॉप पे। अब अभी आपको पूरा प्रेजेंटेबल दिख रहा है ना? ओके नो प्रॉब्लम जस्ट ए मिनट। हाँ बस वही मैं बोला कि अच्छा लगा बहुत दिनों बाद आपको देखे। और ओह ग्रेट। ओके तो YouTube स्ट्रीमिंग लिंक आपने शेयर कर दिया है? नहीं नो इश्यूज, अगर वो है तो यू कैन सी। हाँ आप मेरे को भेज दीजिएगा ना, ठीक है। हाँ मेरे को दिख रही है। राइट। हाँ हम एक ही बैच में किए थे। हाँ हो गया एक्चुअली मेरा भांजा, मैंने उसकी हेल्प लिया, साथ में उसको लिया। अभी मैं दूसरे लैपटॉप से 2 लैपटॉप की हेल्प से कर रहा हूँ। हाँ हो रहा है ना? और ठीक है ना? नो इशू, नो इश्यूज। और बस बढ़िया।
How is life?
लाइफ इज गुड, लिटिल बिट बिजी।
That it will be there. Okay, मैं call cut करता हूँ. We'll talk through this.
ओके ओके।
So right now people are coming slowly. We have 28 participants, and purpose of this presentation is just to students about techniques, correct? No, what we they study and what they. So now our new principal sir is Professor Dr. Raman Dhan sir. You are there now. That Zoom we can see you visible. No issue. Nice to see you after a long time. You are connected.
On है? Right. जल्दी लेना बस।
So I request all participants that please wait for another 2-3 minutes. We will start by 11 or 11:02 maximum, because time is very crucial, and Mr. Gursaranjit Singh has some other schedule also. He has some other work. So sharply 11 or another maximum 2 minutes late, we will start. Please have patience and stay with us. फुल कैमरा कीजिए गुरशरण, थोड़ा अपना आपका। लकड़ी दिख रहा है, अच्छा नहीं लगता। या गुरशरणजीत जी,
We can start, Sujit.
या 1 मिनट। आप पूरे स्क्रीन में नहीं दिख रहे हैं।
मैं नहीं दिख रहा?
दिख रहे हो आप, नॉट फुल। some that option will be there, no, for enlarge.
अभी कैसा है?
Okay. For me, maybe for participants, maybe able to see you on full screen. Okay, so you just share your screen side time being, and in the meantime, I will introduce. We have Dr. K.S. Katagi sir in our group. We have Shivkumar sir. We have so many people are there. Yeah, Guru Sharan. Yeah, nice to see. We got first page, no? Yeah, fine.
Right.
So now we'll start. One second. So, good morning to one and all participants present here. On behalf of our Honorable Chancellor Dr. Prabhakar V. Kore sir, Honorable Vice Chancellor Dr. Vivek A. Sahuji sir, Registrar V. A. Kotiwali sir, Head of the Organizing Committee, Principal of Kelly College of Pharmacy, Dr. Professor Ramandang sir, and the member of this Alumni Association coordinating team, Mr. Yuvraj Singh Surana and Mrs. Rekha Yu. I, Sujit Kumar, on behalf of Kelly Alumni Association, Bangalore, would like to welcome you for this wonderful presentation on the basics of pharmaceutical scale-up and process development by our distinguished alumni, Mr. Gurusaranjit Singh. Before I request Mr. Gurusaranjit Singh to start his presentation, let me have an introduction about Mr. Gurusaranjit Singh. Mr. Gurusaranjit Singh is an alumni of 2004 batch for B Pharma. He completed and then he did his M Pharma in pharmaceutical technology in 2006 to 8 batch from Kelly College of Pharmacy, Bangalore. From 2008 to 2020, he has a very vast experience of production, technology transfer, and he rendered his service in organizations like Biocon, Glenmark, Cipla, and right now he is associated with Dr. Reddy Laboratories as a team leader. So a person with a great personality and humble, with a person with kind attitude and a knowledgeable and a skillful resource person of Kelly Alumni Association, Bangalore. We are really fortunate to have Mr. Gurusaranjit Singh as our member. And with this small introduction, I would like to request Mr. Gurusaranjit Singh to start his presentation. Gurusaranjit sir, please start with presentation.
Thank you, Dr. Sujit. Thanks for giving me the opportunity to interact with the alumni and current students of KLE. So, as you already briefed about myself, and I feel very happy because it's a very proud moment for me to interact with all my college mates, my juniors, and my teachers through this online portal. And today I've selected this topic. The title of the topic is Basics of Pharmaceutical Scale Up and Process Development. So I've selected this topic because this is something which we can say that it is need of the time. And what I personally felt that students actually need awareness on this topic, because I've seen that students interacting with me from KLE, my juniors, they have called me so many times regarding the jobs and opportunities and further careers. So I felt that students need some kind of further more clarity that in which area they want to pursue their career. Because I've seen that people sometimes they interacted with me that we want to join. I was in Biocon Bangalore, so I got calls from so many juniors. So I will share one of the instance that people approached me that we want to join R&D. So when I asked that what you will be doing in R&D, so they were not clear. So then I felt that some basic knowledge about the role in the industry must be clear with the students so that they can make their minds that which area or which field in the industry they should go, and accordingly they can start developing their skills when they are doing their graduation and post graduation. It happened with us also, because I started my career with R&D. I started my career with Cadila Pharmaceutical R&D. I worked with Glenmark R&D, Strides Arco Lab R&D Bangalore, Biocon R&D Bangalore, and currently I am working with Dr. Reddy's Laboratory in MSET department as process lead. So I felt that this such kind of topics will give the clarity to the students to make their objective very much clear, that how they will be pursuing their career. So with this, I will start with the topic scale up and process development. So what is scale up exactly, and what is process development? So scale up, for example, I am having a batch size of 1 lakh tablets, and I am asked to manufacture a batch size of 10 lakh or 20 lakh or 50 lakh, or as per my organizational requirement. So that is scale up. From one scale to another scale we are moving, that is scale up. So scale up is generally defined as the process of increasing the batch size. Scale up of a process can also be viewed as a procedure for applying the same process to different output volumes. And process development. So process development is the process of drug manufacturing and can be broken down into range of unit operations such as blending, granulation, milling, coating, compression, filling, and depending upon the type of dosage form. So then comes the question, why scale up and when scale up? So why scale up? So as I said earlier, that whenever there is a need of the organization, it could be filing purpose, or it could be just to increase the batch size, we will be asked to increase the batch size by doing the scale up. So these are the three basic strategic approaches for the scale up. So first is laboratory scale batches, another one is pilot scale batches, third one is production scale batches. So laboratory scale batches are the batches which are produced at research and early development laboratory scale. They may be very small batch sizes, 100 to 1000 times less than the production scale. Laboratory scale batches may be used to support formulation and packaging development, early clinical and preclinical stages. Pilot batches. Pilot batches are used in the process development and optimization stage. They may be used to support preclinical and mid to later stage clinical evaluation, and also to support formal stability studies. If supporting formal registration, a pilot batch size should correspond to at least 10% of the production scale. For oral solid forms, this size should be generally 10% of the production scale or 1 lakh units, whichever is greater. Then comes our final approach, that is production scale batches. These batches are of the size that will be produced during the routine manufacturing and marketing of the product. Data on production scale batches may not be always available prior to granting marketing authorization.
Gursharanjit sir.
Yes, sir. Please tell me.
Gursharanjit sir.
Hello. Yes,
can you little adjust your camera? Your face is not visible. Little.
Okay.
Yeah,
is it okay?
Yeah, fine. Little. Camera, your face is cut, not full.
Is it okay?
Little more. No, up. We are not able to see you. Yeah, fine. Excellent. Yeah, yes, sir. Yeah, fine. Please.
This is fine, no?
Yeah. Sorry. Please. Can
okay. So for the scale up, similarity is most important. So I have projected two pictures. So do you feel both the pictures are similar? I'm talking about the lady and a puppy on the right hand side. Are they look similar? Okay. So similarity is most important when we are talking about scale up and process development. So I will come on the further slide, which will give you more clarity that what I'm talking about similarity and what is the meaning for the similarity here and how to achieve process similarity during the scale up. So basically, if at all we want to go for a scale up, our process should be similar. Similar in the sense, it should be geometrical similarity, it should be kinematic similarity, and it should be dynamic similarity. Geometric similarity means characteristic linear dimensions. Kinematic similarity is same ratio of velocities between 2 points, and dynamic similarity is same ratio of forces between 2 points. So process similarity can be achieved after getting geometrical similarity, kinematic similarity, and dynamic similarity. So I will further elaborate this in a simple way that what exactly it means. So this picture will give you the clear idea. So if at all we are talking about the geometry, so my equipment, geometrically, if my lab scale or pilot scale equipment or production scale equipment, if they look geometrically similar, then I can say that my geometrical similarity is there. And if kinematically same ratio of forces, if it is observed, if you can see on the picture that both the side arrows are shown, so this shows that the ratio of the velocities between the 2 points is same. And in a similar manner, dynamic similarity, that is same ratio of the forces between the 2 points. If at all I am having all the three kinds of similarities, then I can say that my process is similar. So being similar is the prerequisite for my scale up. And further comes is dimensionless numbers. So dimensionless numbers are the numbers which are representing a property of a physical system but not measured on scale by physical units. So these are the three kinds of dimensionless number I've mentioned. There are many more dimensionless numbers, and it depends upon different kinds of equipment processes. So there are various dimensionless numbers. So in routine, whatever are used, these are power number, Froude number, and a Reynolds number. So in this presentation, basically I will be giving an overview of the scale up for the OSDs, because OSDs are the dosage form which are maximum manufactured, and the knowledge about the OSD scale up is must if anybody is interested to join the industry. So basically in OSD, there are these four basic kind of operations or unit operations, I can say. First is blending process. Second one is granulation process. Third is compression, and fourth is coating process. Blending process is very much critical process, and the basic principle of the blending is diffusion or dispersion, convection or shear. So these are the three basic principles for the blending. And for any oral solid dosage form where the dose is potent or the dose is low, like the dose is less than 25 mg, ensuring the uniformity of the blend is very much important as well as critical. So here in this slide, I have presented blending principle along with the examples of the various kinds of blender which are used in the industry. Diffusion. So basically, what is diffusion? Diffusion is nothing but it is the redistribution of the particles by random motion within the powder blend, which causes them to change their position relative to one another. The example of the blenders which are commonly used in the industry are double cone blender, octagonal blender, V shell blender, and bin blender. Next one is convection. Convection, what happens? The transfer of one location to another is within the mixture with motion that is imparted by impeller as in the ribbon blender. So I mean to say that here the movement of your powder will be with the help of an external support, that is a ribbon. Next is shear. Shear is the change in the configuration of ingredients through the formation of slip planes with the motion imparted by high intensity mixers. The example of this is rapid mixer granulator, where impellers are there, so which will create the slip planes once they will start moving, and which will help in the mixing of the blend. So this is the pictorial view of different blenders which I talked about: double cone blender, octagonal blender, V blender, and Win blender. These all are diffusion blenders. Next is ribbon blender. So a ribbon blender, if you can see, there are number of ribbons, and these ribbons will be moving with the help of a motor, and it will cause the movement of the powder from one place to another. But these kind of blenders nowadays are not used in the industry. They are more frequently used in the bakery industry for making the dough, because there are a lot of dead spots in these, and there are uniformity related issues in the blenders, so these are not much used. Next is shear mixer, and this is a rapid mixer granulator. As I said earlier, it is having impeller blades which will help to create the slip planes in between the powder, and it will cause the movement of the powder, which will ensure the mixing of the powder. Next comes the type of mixer and blender selection. It is very much important. So first of all, at the developmental stage, during formulation development scientist has to ensure that what kind of mixer the particular product will be having. For example, free flowing mixer, these are non-cohesive. Free flowing powders are having desirable features like minimal need of lubricant and effective contact with the dye. They suffer from serious drawback of segregation of individual components in post-mixing process. Blenders such as bin blender and twin shell blender will be used. Second is cohesive mixer. Cohesive mixer exhibit stick slip characteristics and components are not free flowing. The individual particles are repeatedly broken down and allowed to redistribute within the system to ensure a satisfactory mixed product. Blenders such as high shear mixer, twin shell with I bar will be used. So I bar is nothing but these are intensifier bars which will help the mixing in a particular blender, and also we will call these as baffles. These will be having baffles or intensifier bars. Next is ordered mixer. If one of the constituent of the powder mix is added to a fine micronized form, then on mixing, the larger particles may absorb some of the smaller particles on the active sites on their surfaces, and they are held tenaciously. Blenders such as tumbling mixers, cone mixers, high energy, and many types are used. Now comes the segregation. So I am dealing with a blend. If that blend is free flowing, so there is a probability that I must be facing an issue of segregation, which commonly helps and which is the major headache for any formulation development scientist or for any manufacturing science and technology person, because this is the phenomena which is shown by the most of the powders when your drug is very much less in the your formulation, or the formulation is potent. So different types of segregation are: first, during sifting, small particles slip between the larger ones. So in this, what happens if there is a difference between the size of the particle of API as well as my raw material, there will be difference of the bulk densities. So which will cause the segregation. So if we want to control it, the particle size shall be more than, and it shall be in the ratio of 3:1. Mean particle size should be more than 300 micron. Free flowing piles formed throughout the funnels, and major component is more than 3 times minor one, and vibration effects may cause accelerate or may enhance the segregation due to shifting. So these are the potential root causes for this kind of segregation. And the next one is aeration. Aeration or fluidization, which causes fines to travel on the top with the air, and your materials which are bulky, they will be on the base, and the materials which are light, they will be fluidized, and it will come on the surface of the blend. And this basically depends upon the particle size differences and excessive high shear mixing, due to which air will be introduced during blending, and settling effects. Dusting of the fine particles in the air, fines accumulating at the site or the perimeter of the bin. Blend containing a high proportion of fines due to inadequate granulation or use of fine excipients or API will be having this kind of problems. Arcing or rat holes. This is a very common problem which one will face every time whenever there is flow related issues, because the material which is on the side of the hopper or funnel, it will stick, and the material which is in the center, it will start flowing. So it will cause a rat hole. Because of this rat hole, there will be uniformity related issue or the segregation. So basic solution for this kind of problem is that narrow particle size distributions, more cohesiveness among the ingredients, and reduced material handling. So whenever we have to handle the material, we have to unload it. Unloading shall be done using the chutes. Unloading chutes are available, SS chutes are available, and scooping shall be done very much carefully. There should not be any hammering, and a lot of precaution shall be taken during the transportation. Change in the equipment design, so like angles, vents, cone in bin. So these are the mechanical modification or engineering related modification which could be implemented in the design of the equipment, which will support and which will reduce the chances of the segregation. So equal proportion if possible. So the mix should have 50-50. If we are using a ratio of 50-50, then there is minimal chances that we may face a problem of segregation. So next is scale up parameters. Scale up parameters are very much important, and during the design and during the developmental phase, we have to work upon these scale up parameters very much carefully. So how it will be done? First of all, if at all I want to prepare a batch size of 100 kg from 10 kg, so I have to look about that what is the batch size I will be keeping exactly. So how much kg I have to put in the container or in the blender, that shall be evaluated based upon the volume of the container as well as the bulk density of the product. Bulk density is very much important because every product varies in its nature because of bulk density, particle size, and a lot of other parameters. So first of all, we have to select very carefully that what will be my batch size. Second most important thing in the blender will be blender speed and blending time. So blender speed we have to select in a such a way that it ensures that at particular speed and with that particular blending time we are able to achieve uniformity. So for example, if at all I'm having a blender, so there are different sizes of blender which are used at R&D scale and as well at commercial scale. Blenders of 20 to 50 liter, 250 liter, 500 liters, 500 liters, these are generally 5 liter to 50 liter are used at R&D scale. Blender of 100, 125 to 50, these are generally used at pilot scale. And blenders of 500,000, 2000 and 4000 liter, these are the blender which are used at commercial scales. So the typical RPMs, so RPM or blender speed, it is very much important. I'm reiterating it. So typical RPM for 20 to 50 liter blender is 25 to 30. For 250 liter, typical RPM is 22 to 28 RPM, and for 500 liters, it is 12 to 18 RPM, and for 2000 liter, generally it is 8 to 12 RPM. So, as I explained, as the size of the blender increases, the RPM decreases. So smaller blenders will operate at higher speed and the blender which are of bigger size will be operating at lower speed. And accordingly, we have to, if my geometrical similarity is there, so first of all, our prerequisite will be matching the tip speed and total number of revolutions. So these are the two criteria how blending time and blending speed will be finalized. So tip speed is equivalent to tip speed means the formula for the tip speed is 2 pi into r into capital R. So r is basically h, r is equal to h by 2. H is the height of the blender and r is the RPM of the blender. So tip speed shall be matched and keeping the number of revolution constant by varying the time. And second is total number of revolutions. So what is RPM? RPM is the revolution per minute. So according to that, we have to calculate that how many revolution my blender is taking. So according to that, I have to fix my number of revolutions. So we will be matching the number of revolutions. Next comes the compression process. So prerequisite for compression is compatibility and fluidity. So here I have summarized just common advantages of the compressed dosage form. These are accurate, stable, and with least variability, cheapest dosage form, and elegant, prolonged stability, great patient compliance, most stable with respect to physical, chemical, and microbial attributes, low manufacturing cost, and ease of administration. So the type of the compression machine. So different kinds of compression machines are used in the industry. They are single rotary compression machine, double rotary compression machine, and nowadays fully automated compression machines from different manufacturer are available, and which where we can study a lot of things in detail. A lot of data can be generated, and here on the further table I have explained about the type of the compression tooling. So different types of tooling which are used during the compression, it is B tooling, D tooling, BB tooling, and DB tooling. So this kind of tooling is on the basis upon punch diameter, die diameter, and maximum size of the tablet. So if at all we want to select a tooling, we have to consider what kind of hardness, what kind of tablet weight is my requirement. Accordingly, the particular type of tooling shall be selected. There seems to be some problem. I'm not able to project the next slide. It is showing something black. So no issues. I will explain that. So in this screen, I have presented the different kind of punches that are used in the industry. So as I said, that selection of the tooling basically would be depending upon the type of the dosage form. If at all, if it is a tablet, then question comes that what kind of tablet it is, whether it is a flat tablet, whether it is a shallow concave tablets, it is a deep concave tablet, it's a extra deep concave tablets, or whether it is a modified tablet or a flat face bevel edges tablet. So according to that, tooling shall be selected. Uh, Sujit, are you able to see the screen? Because I am seeing that.
No, it's blank.
Yeah, I don't know. It seems to be some technical issue.
It's okay.
Because,
continue.
yeah, while presenting, I'm not able to see it, but I am able to see it on my laptop.
Yeah, here also we are not able to see. Yeah, you can continue.
Okay, no issues. I will explain. And next, I have explained about the different kind of shapes of the tooling which are available. So it is convex cup shaped, or compound cup shaped, or capsule shaped, or oval shaped. So this is what I have projected, but I'm sorry, you may not be able to see.
Yeah, now it's coming.
Yeah, actually these were the pictorials which I thought that it will be very much helpful for the students, so they can have an actual look. So I collected from various resources, but unfortunately I'm not able to project them. Anyway, so I will come to the next. This is the next I've projected about the composition of the tooling material. So friends, it's very much important that whenever a process is designed and a tooling is selected, so basically different kinds of materials are used for the manufacturing of the toolings, like S1, S7, D2. So these are the particular kind of steels which are used, and these grades of steel will be selected according to the nature of the API or the raw material which are going to be used for particular material. Because sometimes what happens, if you are selecting a material, for example, if it is a material which will cause the corrosion or rusting, so then it will be difficult to have a common type of tooling. So according to that, we have to give our request to our tooling vendor. So we have to get the proposal from the vendor that what kind of steels I can use, and also the kind of MOC which we are requesting to the vendor. It will depend upon the kind of hardness which is required for my compression, because it will depend upon the compressibility of the particular formula or formulation. Some are having very low compressibility, some are having very good compressibility. According to that, our request shall be given to the vendor. Accordingly, the tooling shall be procured. It is very much important. I am reiterating that. So next is scale up of the compression process. During the scale up of the compression process, it is very much important uh, to have a proper compaction, compression, and consolidation. Compaction is the compression and consolidation of two phases, that is particular solid and the gas system, by the application of an external force. Next is compression. Compression causes an increase in the apparent density or reduction in volume by displacement of the air. So whatever the air is entrapped in between the granule, so that will be displaced when we will apply certain kind of force or pressure on that while compacting. Consolidation is defined as an increase in the mechanical strength due to particle-particle interaction. So once compaction happens, the particle-particle interaction will be increased, which will give the mechanical strength, and our tablets will be formed. Further are the few important terms which are used during the compression. So out of them, the most critical and important is what I feel is that dwell time. So I will explain about the dwell time. So rest of the terms you can go through. For example, this is feeding time, the time when the dye is fed with the powder, consolidation or solidification time, the portion of the contact time when punches are changing their vertical position in reference to the roll, decreasing the distance between their tips. Next is dwell time. Dwell time is the portion of the contact time when punches are not changing their vertical position in reference to the compression roller. Next is decompression or relaxation time. The portion of contact time when punches are changing their vertical position in reference to the rolls, increasing the distance between their tips before losing a contact with the rolls. In the previous slide, I have presented a GIF. So unfortunately, I'm not able to show. So I thought of showing you that and correlating these terms with that image. But I have to explain these. So compression time. So compression time is the portion of the contact time before the decompression period begins. Contact time is the time when both punches are moving and having their tips in contact with the material that is being compacted and their heads are in contact with the pressure rolls. Ejection time, the time when the tablet is being ejected from the die. Total time, the time required to produce one tablet on a press. Feeding efficiency factor. Feeding efficiency factor is that is denoted by EF. It depends upon the TF divided by TT. TF is my total feeding time and TT is my total time which is required to form one tablet. So next is consolidation efficiency factor, that is ES. It depends upon TS, that is consolidation time, divided by TT, that is total time. Next is efficiency, ejection efficiency factor. Ejection efficiency factor is TE. TE is your ejection time and divided by TT. TT is our total time to form one tablet. So here I've projected the different kinds of cams. So during the compression, before going to compression, it is very important to select particular cam. What kind of cam will be used during my compression? So this is a major challenge. So I have defined it along with the weights, weight of the tablet, what is the weight of the tablet, and what kind of cam shall we use. So I have explained here two kinds of tablet tooling from different make. One is from the Sejong and another is from the Cadmac. So this is how the cam selection will be done. And nowadays the toolings or the machineries which are coming, automated machine. So in automated machines, we will be getting the option of auto selection of the, so option will be given by the machine itself that what kind of cam shall be selected. So next is dwell time. We have already talked about the dwell time. Dwell time is very much important, and dwell time depends upon the head flat and as well as on the number of punches, pitch circle diameter and tablet per hours. So scale up during wet granulation using high shear or RMG. So during the granulation, the primary powder particles are made adhered to form larger multi-particle entities. In other words, the process of collecting the particle together with the help of bond formation with the help of binders. So binder will help the powders to come closer and come in close proximity, which is known as granules. So the benefits of the granules are improved flow properties, reduced segregation, better content uniformity, reduced dust generation, increased density and lower packed volumes, improved properties during compression. So again, so I've presented here stages of wet granulation, and it's not coming. So the stages of the granulation are: first is pendular stage, where binder will be poured on the blend, and it will start forming slight smaller bridges between the blend particles. So this is just the initial stages, and further there will be more addition of, and these bonds will be increased more. Then this stage will be known as funicular stage. Funicular stage is intermediary stage, and the final stage where the granules will be formed, it will be capillary stage. So at capillary stage, most of the binder will come in contact with the materials, or it will binder will accommodate itself in between the granules and the powder, whatever is there. So this is my endpoint where we should stop our granulation. But after this, there is one more phase. Being a formulator or being a scientist, we should avoid that stage. That stage is known as droplet stage. In the droplet stage, if at all we are not able to stop our process once the endpoint is achieved, then if mixing is continued or additional quantity of binder, it could be water or it could be isopropyl alcohol. So if at all it is added, then it will form a droplet. The droplet will form a lumpy mass, and our batch will be spoiled. So we have to take very much attentive or care during the granulation, that we should not reach this stage. We should evaluate proper endpoint of the granulation. The proper granulation endpoint is achieved by recording proper amperages of my impeller, amperages of my chopper, which will give a proper endpoint and a proper product. So on the next stage, I've explained about the flowability and compressibility. Here again, you're not able to see. I'm very sorry for that. Flow characteristics are very much important for a granular material, because if the flow is not proper from the hopper or in the feed frame, there is segregation on the turret, that is very much important parameter. So ideal flow characteristics involve: angle of repose should be good. So good is called 31 to 35 angle of repose, it is good. Excellent is 25 to 30. So we have to achieve the excellent or good, so to have a product which is running smoothly on the commercial scale. Compressibility index should be in the range of 11 to 15, which is called GIRD, and it should be more than 11, and which is called excellent. And Hausner ratio should be in the range of 1 to 1.18, so which is the prerequisite to have a good product. So key points of wet granulation optimization. So if at all we have to optimize my process, what we should do: type and quantity of the binder. So as I mentioned in the previous step, that we have to avoid overwetting. So how I can avoid overwetting? So we have to fix the quantity of the binder, type and quantity of the binder, characteristic of the drug and other excipients. If at all my drug is freely soluble in water, and I'm using a binder which is having freely solubility into the water, then it will affect my endpoint. And the diluents, for example lactose is freely soluble in water, but apart from that, MCC is not freely soluble in water. So there will be difference in the granulation using MCC and lactose. So the formulator or the scientist should evaluate these things during his scale up studies. Spreading of the binder, temperature and viscosity. So for example, if at all we are manufacturing a product with a granulation using maize starch paste, if at all the consistency of the or viscosity of the binder is not fixed, so how the viscosity will be controlled? With the temperature. So maize starch cooking is very much important. At what temperature maize starch becomes fully cooked, at that point or that point of the time we should add the binder. Okay. Next is mixing time, process, other process variables, apparatus variables, impeller movements. This will also play very much important role in the wet granulation. So next point comes key important factor for scale up of wet granulation. First, as I said earlier, geometrical similarity. So in this, the important point will be the percentage occupancy. We should maintain an occupancy of the dry mix across all the scales to achieve a geometrical similarity. Powder bed to RMG diameter ratio. This is also very much important that it is properly evaluated. And third one is relative impeller swept volume. So to achieve the geometrical similarity, these three things are very much important. Relative swept diameter is the volume swept by the impeller for one revolution. This can be represented as the ratio of the impeller height and powder bed. So the impeller swept volume is equivalent to H by H. So H is impeller height, capital H is impeller height, and divided by H. H is the height of the powder bed. Next comes the kinematic and dynamic similarities. So to achieve the kinematic and dynamic similarities, so we have to achieve the tip speed. So to achieve the tip speed, we have to evaluate both the RMGs at my lab scale or RMGs at my pilot scale and the RMGs at the commercial scale. So we have to maintain the constant tip speed. Okay, if at all we are maintaining this constant tip speed, we can anticipate that the kind of granulation which will be achieved will be uniform or will be comparable at both the scales. For achieving the geometrical similarity at different scales, this is a small flow chart. So first of all, the equipment is having same principle of operation. No, then scale up is not possible. If at all principle of operation is same, then we can proceed further. And if equipment are geometrically similar with respect to all dimension and angles, so then scale up of the particular batch size along with the particular occupancy is possible. If they are not geometrically similar, scale up of the batch size by matching the H by D ratio of the or the impeller swept volume to maintain the geometrical similarity has to be done. So here in this slide, I have tried to present the linkage of the product with the process. So our ultimate goal is to send a very good product that is safe, that is effective, and it should be in the hand of the patient. So for that, what we need to do, being a formulation scientist or being a process scientist or being a process technologist, what is our role? The critical material attribute shall be controlled. The critical process parameter shall be in control. If at all my critical material attributes and my critical process parameters are in control, then our product will be having controlled critical quality attributes. If at all my critical quality attributes are in control, the product will be having a good safety as well as efficacy. So I mean to say that the outcome, clinical outcome, will depend upon whatever CQA, CPP, and CMAs are controlled during the commercial scale or during the manufacture. So understanding of the CMA, CPPs, and CQA, it's very important. So what is critical quality attributes? So I'm talking about that these three things are very much important to have a very good product. So exactly what is that? So critical quality attribute, it is a physical, chemical, biological, or microbial property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. On the other words, critical quality attributes at the time of the release of the batch, what should be my SA, what should be my dissolution, all these things, how much should be the percentage of the impurities that should be present in my product at the time of the release. All these are the critical quality attributes. Next is critical process parameters. Critical process parameter is the process parameter whose variability has an impact on a CQA and therefore should be monitored or controlled to ensure the process that produces the desired quality. Next comes the critical material attribute, a physical, chemical, biological, microbial property or characteristic of the input material that should be within an appropriate limit or range or distribution to ensure the desired quality of product or material output. So here again I have presented the relationship between all the threes. So first of all, in the first there are CMAs of the input material. Next is my CPP. So I'm giving appropriate CMAs. Then I have to control my CPPs, and in turn I will be getting CQAs or the output material that will be complying to the final quality of the product. So this is the example which shows the approach to identify material attributes and process parameters. So a combination of perfectly designed process with control on CMA and CPP will be able to deliver the product within the predefined CQAs. So first step, if you will see, it is identify drug product CQAs. Second step is for each process, identify intermediate CQAs that impact drug product CQAs. Step 3 is identify material attributes and process parameters that may impact the intermediate CQAs of the process step. So for example, my drug product CQA is content uniformity. So content uniformity is very much important when my product is potent. Means my label claim in the formulation or in the tablet is it is less than 25 mg. I'm having a label claim of 1 mg or 2 mg. Then it is very much important to ensure a proper content uniformity. So for that, intermediate CQA will be very much important. So what will be my intermediate CQA? My intermediate CQA will be blend uniformity. So during blending stage, we have to ensure that my blend is uniform when sampled from the different location of the blender. So once that my intermediate CQA is under control, obviously my CQA, final CQA, that is content uniformity. Generally, content uniformity is performed on the stratified sample. Whole compression run is divided into a number of statas and the sample shall be collected from those statas. अगर मेरा कंप्रेशन 30 घंटे चलना है तो मैं अपने पूरे प्रोसेस को 30 घंटे में डिवाइड कर लूंगा. आई विल ब्रेक द होल कंप्रेशन प्रोसेस इनटू 30 स्टेटास. सो एवरी आवर I will be collecting a certain number of tablets, and once the compression is complete, for example 30 hours compression is complete, I am having 30 statas. So those all the 30 statas shall be analyzed and those 30 statas should comply. Okay, so once those comply, it will give the confidence level so that during the compression run, throughout the compression run, my drug is uniformly distributed into the unit dosage form, that is my tablet. So the process variable for that will be having a better or a good blend uniformity. What process variable will be important? First is particle size distribution. So how my particle size distribution is, that is very much important. Next is loading level. If at all I am loading my granules in a blender or in a RMG while I'm doing granulation, my occupancy is more than 85%. मेरा एक ब्लेंडर है उसमें मैंने 90% मटेरियल डाल दिया, only 10% space is left for the material movement and I will be moving the blender. So it will not give the proper uniformity. It will not help the blend to move inside the blender. So it is very much important to ensure the loading level and as well as the number of revolution. So the number of revolution, as I explained at the stage of blending, that it is very much important to finalize the number of revolution or the blending time. So once that blending time or the number of the revolutions are achieved, we can say that particular blend will be having uniformity and it will give the confidence level on the subsequent stages. Next is identifying the potentially high risk material attributes or my process parameters. So first column is showing input materials. Input materials is having particle size distribution and particle size. So we have already briefly discussed on that. So the purpose of here I'm presenting it that we are identifying potentially high risk. So these are very much high risk. My material attributes, different material attributes are there if you see. So that is particle size distribution, fines or oversize, particle shape, blend or tap density, cohesive or adhesive properties, electrostatic properties, and moisture content. But if at all I'm focusing my CQA is by blend uniformity. My quality attribute is my blend uniformity. So for that, what is most important? Mixer load level or the number of revolution, and also environmental and temperature and RH. That is not very much critical, but it depends upon product to product. If at all my RH is very much high, there are chances that my material may grab moisture from the environment while manufacturing, and it may show sticking or adhesion to the walls of the manufacturing vessels. And here is the summary of the. No, it's not coming proper. So here is the summary of the material attributes and the process parameters. So stepwise, I have divided a compression OST process. I have bifurcated, and the unit operation are divided. For example, first of all, it is blending. So in the blending stage, the parameter will be drug substance particle size and bin load and blender revolutions. And my measured attributes will be uniformity of the dosage form. So different kinds of studies are performed to finalize these material attributes, critical quality attributes, and CPPs. So on the basis of all these, controlled strategy is designed. Next, if you are able to see, it is milling. This is a milling screen, and milling speed are my parameters. Third one is lubrication of the blend, where blender revolution will also impact the uniformity of the dosage form. Sixth unit operation is compression, where parameters will be compression force, dwell time. So these will affect uniformity of the dosage form, tablet core hardness, tablet core friability, tablet weight variation, tablet core disintegration, tablet core dissolution, punch sticking. And next is film coating. Film coating, the parameters will be pan load, spray rate, temperature of the exhaust air or the inlet air, coating weight gain. So the measured attributes will be tablet appearance and tablet water content. So on the basis of all these studies, control strategy shall be provided, and on the basis of all the studies and this control strategy, validation shall be planned. And if the validation, during the validation, if at all I am doing a blending step, and I've already assigned that how much blend revolution will be there, or how much blending time will be there, or at milling what kind of screen shall be used, and what will be the speed of the impeller. If it is intragranular lubrication, what is percentage of the bin loading, what is the revolution of the blender. If it is dry granulation stage or milling, roller force, how much is the roller force, how much is the gap width, how much is the mill screen, how much is the roll speed. If it is extra granular lubrication, percentage of the bin loading or blend revolutions. During compression, how much is the compression force based upon the tablet hardness, compression speed, dwell time. And if it is film coating, how much is the percentage of the pan fill, how much is the exhaust air temperature, exhaust air humidity, and coating weight. So these, all these controls will be able to control my CQAs, and basis upon this control strategy we can say that using these particular parameters, particular material, and particular process, we can develop a product which can give the output as per the predefined specification. Or in the other words, I can say that my product will be having all the quality and quality, safety, and quality, safety, and efficacy as per the predefined specification, if at all these things are met. So this is how a process is developed and how a scale up is done. This topic, particular topic, it is a very vast topic, and being students, I thought of giving you glimpse of each and every stage. So I've tried, actually there was shortage of time, and I was not able to accommodate much more material, which I thought maybe some other times I will try to have all those things, and I will try to explain further things to all of you, so that you are having a better idea that how a process is developed and how scale up is done at the industrial scale or at a commercial scale. These are the references which I've taken from FDA, as well as from pharmaceutical dosage form of tablets by Larry L, and developing solid oral dosage forms by Yong Kui, pharmaceutical process scale up by Michael Levin, and ICH pharmaceutical development Q8. And I will suggest, if at all you are keen to learn more and more in this, because this scale up is a very critical task, and it involves a lot of studies, a lot of calculation. So my personal recommendation will be that if at all you are interested, you go through this book, Developing Solid Oral Dosage Forms, and once you will go through this, then try to read second book, a third book which is mentioned, that is Pharmaceutical Process Scale Up by Michael Levin. So it will give you very good picture, if at all you're planning to pursue a career in manufacturing science and technology, or technology transfer or formulation development. So these are my personal recommendations, and I will request you, if at all, if you're having any query or question, you're free to ask. So I will try to answer your questions.
Okay.
Yeah, Dr. Sujit,
Yeah,
please.
thank you, Guru Sharanjit Singh ji. Now, please you can stop sharing your screen, no issue. And really, it was a very wonderful presentation, very informative, and it reminded me just like I was sitting in a industrial pharmacy class. It is a quite a type of lecture, and I find myself it will be very informative for the students. Now, it is the a term for participants. If they have any queries, any query, any doubt, they can raise their hand and they can ask the questions. So we'll take few questions from this presentation. If participants have, please raise their hand and ask it. And Mr. Gusaranjit Singh ji will answer your question. So we have one question from Mr. Madhusudan Reddy, and I request
Okay.
Madhusudan Reddy sir to unmute yourself and ask the question, sir. Mr. Madhusudan Reddy, please unmute yourself and ask the question. Yes, sir. Mr. Madhusudan Reddy. Yeah, please, you can ask the question, Mr. Madhusudan Reddy. Yeah, Madhusudan sir, are you listening? Hello. Hello. Okay. So any next participants, anyone has any queries? Any query, doubts, questions, please clarify. Mr. I have one doubt from Maruti. Please unmute yourself, Maruti, and ask the question.
Hello, sir. I have one question.
Yeah,
Yeah,
please.
please.
So my question is, what are the parameters? What are the mathematical equations to be considered for this scale-up of compression process? Means from less station compression machine to more compression machines to, or from single rotary compression machine to double rotary compression machine. So what could be the process parameters to be considered for the scale-up process? This is my question.
Okay. So, first of all, for any kind of scale up, as I mentioned, that we have to focus on that my make, model, design, and working principle that should be similar. Okay. If at all it is not matching, then we have to generate the data. The evaluation shall be done. Accordingly, we have to calculate that what is the dwell time. Okay. If at all I'm currently running the machine at a 30 rpm, if at all I'm going ahead for a higher station machine, that is different. So whether the tooling, same kind of tooling will be feasible with that. In that we will be having, if at all the tooling is same, then it is okay. If it is there is a difference in the tooling, for example there is a difference in the punch size, so obviously it will be affecting my dwell time. So we have to match the dwell time, we have to perform the dwell time calculation, that how much dwell time we have to keep. So this is how we will be moving ahead for the scale up of the compression machines.
Yeah, I think you got the answer, Mr. Maruti. So we have some more queries. One from Shakti Singh Rathod. I will request Shakti Singh Rathod to unmute and ask the question. Yes, please go ahead, Mr. Shakti Singh Rathod.
First of all, thanks a lot to Guru Sanjit ji for a very nice informative session. This is delivered to an industry, to academic institution, and it's a really very glimpse of presentation that you have taught to the student and the alumni. And so my question is like that: if you are going for the scale up from R&D to bigger scale up pilot plant, and from a pilot plant to if you are going for the manufacturing scale.
Right.
So you have spoken about certain thing in granulations and blending, and also you have spoke about compression. So likewise, you have said that the basic principle of this scale up is your make and model of the instrument should be same at every stage. But
Yes.
realistically, this is not happen because most of the time we don't have that liberty to match all the equipment like to like,
Right.
and we have certain model find differently at different pilot scale and manufacturing scale.
Right.
So on that time, suppose to matching the tip speed and all this matching for impeller dia and at the
Yes.
blending stage, matching all your tip speed and the matching that brim volume in the coating stage. So that is a very tedious task. So in that time, suppose if I'm going for a very different make and model of RMG, where my impeller and chopper geometry is not same. Instead, that if you have very different geometry, and the working principle that are same, but the geometry will be different, like in RMG and in Blender. So can you just explain your experience in those kind of challenges where you have faced where that the working principle are not same, and you have to go with that scale up due to unavailability of that same make and model. Yes, Gurujinji, can you just explain me
Yeah,
on
yeah,
that?
sure. So first of all, as I mentioned that uh, we have to uh, try that we are having geometrical similarity at the same time we have to achieve the kinematic similarity. If at all geometric similarity is not there, then obviously we have to focus on kinematic and dynamic similarities, and that is achieved first is by impeller tip speed. Okay, we are matching the tip speed and distance traveled by the granules, right? So these are the two basic things we are criteria which we will try to fulfill. And apart from that, if at all we are totally the design which is not matching, then sufficient data need to be generated. For example, at one RMG we are getting one endpoint, one that impeller amperage I am getting a 30. Okay, and the next RMG which you will not be having the same, in that personally we need to monitor and we have to get the endpoint, like the what are the basic criteria for the getting the endpoint. So endpoint like generally how it is done, one is amperage based, second is time based. So in that case we have to finalize the granulation amperage by time base, how much time I should give. So that will be based upon the trial. During the trial we need to generate the data that how much if at all I am, my mixing time is 3 minutes and my binder addition time is 2 minutes and my further mixing is 3 minutes and overall process is of 10 minutes. So this is, and at these levels if at all I am using this particular time and my endpoint is achieved, so that will depend upon the physical observation. So I've monitored the granulation and I got that this is my endpoint, so that shall be challenged. First will be trial batch, and during the subsequent trials that will be my CB batches. CB batches these once I've gained the knowledge from the trial batch, I will be challenging it on the commercial the exactly validation scale level. So if at all my batch size is 50 kg, so I have taken a trial with the 50 kg, my CB batch size will be further 50 kg, and also my validation batch size is also 50 kg. So I have gained a knowledge on the trial batch of 50 kg. So I am having the ranges now. So I have set the limit that this will be my binder addition time and all those things during the trial. I will challenge those during my CB batches, and I will challenge the outcome. So once that is found, basis upon that, we will assign the ranges. So we will assign the ranges. For example, during the second, in our second process, we got that my amperage is 30 in this particular equipment. So I have to give a range of 25 to 30, and this range shall be challenged during our validation. So if at all with this validation, this given range, we are able to get the desired product, so our process shall be validated. This is how we will be proceeding ahead.
Yeah, definitely. I think what you have said that for the scale up, I think we need to have first similar make and model. But I
Yes.
think if suppose we most of time we don't have that liberty in place, and we have getting all the equipment of different make and capacity. So in that time, I think we should roll out all this thing by taking the trial batch or engineering batch at our industrial scale or at pilot scale. And
Yes.
based on that, because a lot of time we don't have that thing, like we don't have the matching capacity, we don't have the similar
Yes.
geometry in the
Exactly.
RNG blender, and also the drying is also playing some crucial role while we are going for some product which are more sensitive for the granulation. So in that time, I think we just need to have all the calculation, like matching all the bed volume, expansion volume. So I think that
Yeah,
point also
yes.
we need to
Yes,
incorporate.
by
And
maintaining how much is my H by D ratios. So these calculation, and at the same time, number of trials will be more. If at all our similarity is not there, then the risk will be more. So and we have to generate more and more data. I will give you an example. I was in Biocon at that time, in Biocon Bangalore. We have a product which we have developed for US market. So that product was validated, and we started dispatching that formulation from our plant. And at the same time, we do have a challenge that we have to, if I remember, that batch size was 50 lakh tablets for US market. And we got the opportunity to send the same product for Dubai and other emerging markets. And that batch size was only 10 lakh. And we thought of doing the scale up, but the type of the equipment which we were having, those were totally different. So there were limited small scale equipment, like we have to select the kind of blenders which were not exactly matchable, or the principle was not exactly, those were having the intensifier bars, like octagonal blenders we have. So at that time, sufficient data was generated, and basis upon that, by the frequent or repeated trials, we have gone ahead. So this is
yeah, absolutely. So Guru Saranji ji, if time allow, I just speak my experience also. I'm currently working in the same department like MSNT in GlaxoSmithKline.
okay.
And
In Bangalore you're working.
it is I'm working in Mumbai.
Okay, oh, great, good to know.
Likewise, we have technical risk assessment. One documentation is we are
Yes.
globally rolled out for any product which is supposed to be transferred from either from R&D to scale up or to commercial, or from either one side to different sides. So on that time, we have technical risk assessment document is placed. So where whatever CME, CPP, ICQ that you have spoken about, we identify for each different product which is supposed to be transferred. So
Right.
likewise, CPP, ICQ, we have put those cause CQA. So we have established some relationship with ICQA and CPP, and we are scoring that in FMEA. So that is
Yes.
failure mode of analysis. And based on that, we will summarize our CPP, and we are scoring those all risk, and we have mitigation plan in place. And according to that, we have designed one process control strategy. So that has to be
Okay.
validated in our batch document, and we are also validating PCS through validation batch. So I think the time.
Sorry to interrupt, Shakti Singh Rathorji and Gursaran Sirji. Actually, we have more and more queries, and we are running
Yeah,
out of
sorry,
time.
actually the time
I'm
crunch is
sorry.
I will
sorry.
just leave it
Yeah.
at this stage. Thank
Yeah,
you.
you can
No
discuss
issues.
the
It was,
research.
yeah,
you can.
it was a nice interaction. So
Yeah. I'm
if you
sorry.
are having any further doubt, I have
Yeah.
mentioned my mobile number. So
So
if you have any further queries, or if you want to have any
we have
technical
some
discussion,
more
please reach me.
queries. We have some more queries. We'll take another 2 or 3 questions, then we'll wind up the sessions. And later I will request the participants, if they have any doubt, they can contact me. So we have one questions from Mr. Yusufuddin. Yes, Yusufuddin, please ask your questions, please. Yusufuddin. Mr. Yusufuddin, are you listening me? Okay. We have next question from Dr. Anusuya Patil from Kelly College of Pharmacy, Bangalore. Anusuya, madam, please unmute yourself and ask the question. Dr. Anusuya Patil.
Yes, sir.
Yeah,
Am I
please
audible?
go ahead. Yeah, yes, madam, please.
Yeah. Good morning, everyone. I would like to thank Mr. Singh for wonderful presentation. Sir, my question is based on the liquid dosage form. If it is a cough syrup or elixir, when the R&D to scale up, what parameters we should look into it? Can you a little bit tell about the liquid dosage form?
Okay. Fine. So first of all, for the liquid dosage forms, the most important thing will be that whatever the API, it gets dissolved uniformly. So clarity of the syrup or solution is very much important. If it is a solution form, if it is a suspension, then the redispersibility of the suspension, it is very much important. In that case, the important parameters will be that my agitation times, okay, my RPMs, which will we will be keeping for mixing of all those, and especially the problems which I have personally faced. So I will share that. We don't face much problem during syrups, because in syrups the materials all whatever the sugar syrup is used, and API that is freely soluble, and only thing is, if at all it is required, we have to adjust the pH, and apart from that, there are no major challenges, and flavor and other things we are adding. Yes, we do have challenges in case of suspensions. So suspensions are a little bit more critical, and that need to be taken care, and more will be that the kind of the impeller use. So it will ensure the uniformity of the suspension, and at the same time in the formulation, what are the suspending agents are used, so that the flocculation could be avoided. In Glenmark, I was there. We do have faced issues in the marketed product where the suspension formed as solid floccules, and the product came back from the market, and again the work was done on increasing the redispersibility of the that suspension, and the addition of the flocculating agents, further flocculating agents were added. So these were the certain kind of challenges which I personally had. So hope I'm able to answer your question, or still you are having
Yes,
any...
sir. Is there, okay, thank you. Sir,
Yeah,
is there any batch size limit? Suppose antacid in R&D level, I might prepare half liter,
Okay.
I mean to say 500 ml. So
Yes.
is there any capacity limit in scale up? Can I go for 50 liters or 200 liters? Is there any limit? Or we can go for, we can jump directly?
No, if at all it is a solution, so it depends upon the formulation to formulation. So and if at all there are scales. So generally scale up is done from 1x to 10x. So worldwide, this is the strategy which is adopted. For example, if at all we have taken a pilot batches or initial batches at a 1 lakh, so 1 lakh batch size, I'm talking about tablets, then we can go ahead up to 10x. Okay, means 10 lakh tablets we can go. So this is the strategy. If at all we are going beyond that, so there are certain kind of regulatory guidelines. Then we have to justify, and we have to generate additional data for that. If at all it is for domestic market, as such there is no restriction, and only goal is that our ultimate CQAs should be matched. And what is our goal? For example, if at all I am having a 5-liter batch of a particular solution or a syrup, I can go ahead for 50,000 liter also. I can go ahead for further batch size also, if the requirement is there. But for the regulated market, it should be within 1x to 10x. For example, my initial batch size, if it was 500 liter, then I can go ahead for 50,000 liter, 1x to 10x.
Okay, sir. Thank you so much.
Yeah, thank you so much, ma'am. Even you taught us. I was your student that time, and glad to have an interaction with you. It's my pleasure. Thank you so much,
yeah,
ma'am.
Really happy to see you in this. You're working in Dr. Reddy's. Yeah, good.
It's always pleasant to hear from our teachers. So the last question from this session is from Dr. K.S. Katagi. Sir is waiting from long time. Yes, sir. Please ask your question.
Sir, very good morning, and it is a very insightful talk by Singh sir.
Good morning, sir. Please ask.
Sir, my question is, I am working in organic synthesis. I am a research guide in Karnataka University, and we are conducting some synthetic experiments, research experiments, using a micro oven. Hello?
Yes, sir.
Am I audible?
Yes, please go.
The products which are obtained by this micro technology, those cannot be scaled up in industrial level.
Okay.
And these products and time consumption for these particular procedures are very less, and yield is also very nice. But we are not able to scale up these in the industrial level.
Okay.
Okay, sir. Khatge sir, I think you are talking about the API production, bulk synthesis, right?
Yeah, yeah.
Yeah, yeah. So I think the what Gursaranjit Singh sir is all about the scale up of formulations. I think we are, even I am from medicinal chemistry background,
No,
so something like that. Gursaranjit Singh
no,
can answer.
no issues. Sujit, I can answer
Yeah,
because I worked in R&D for near about
fine,
10
fine.
years. So
Great.
formulation experience, whatever I'm having, I will try to answer his question. So could you just brief me that what kind of synthesis or molecule synthesis you are working on currently, and your query about which molecule? So I will try to give my
Organic
input on
synthesis.
that.
Sir, organic synthesis. Usually
Organic
we are setting up some new schemes for novel drug molecules.
Okay, okay,
So we are conducting some experiments in the lab.
okay,
To get the products, we are using the microwave oven also.
okay,
When we use the microwave oven, the time taken by that particular procedure is very less, and
okay.
percentage yield is also good. But this we cannot take into the industry level.
Why? What is the concern you are facing?
So scale up problem.
Scale up.
Yeah.
So for the microwave which you are using, the
Yeah.
similar kind of microwave is
Yeah.
available in the industry.
Yeah, no, it is not available. That's
It is
my
not
question.
available. Okay, that is not available. So
Yeah.
my suggestion will be that just try to explore that what kind of alternatives could be used. What kind of alternative could be used? So I showed interest in your question. Why? Because one of our colleague, he was my senior. So Dr. Gurpal, he is working in Punjab University, Chandigarh. So even I'm staying in Chandigarh only. So he is working on the synthesis part also. He is in touch with the number of projects and number of people those who are working in a similar area. So no issues. You can have my number. We can have offline discussion, and I will try to support you. I can connect with you with Mr. Gurpal. So I will try that your product gets scaled up.
Thank you. Thank
Yeah,
you, sir.
yeah. You
Thank
can
you.
share me the details so we can discuss. Not an issue.
Sure, sure, sure.
Yeah.
Thank you very
Thank
much,
you
sir.
so much. Thank you. Any other question, please?
Kumar sir, please ask the question.
Yeah, Gursharan, you are able to hear me?
Yes, sir. Good
How are
afternoon,
you?
sir. I'm good, sir. How are you?
Yeah, very nice to see you.
Yeah,
Really,
same here,
in
sir.
fact.
Same here. Thank you, sir.
Yeah, I had a doubt because I was in industry long back, maybe around 10 years back,
Okay,
and
sir.
the scaling of granulation, for example.
Right, sir. Right, sir.
What are the critical issues? For example, as I know, in small scale, what they do is they determine the end point by a different method, what I can know. And
Yes.
in the large scale, what they do is they go for an electrical method, something
Right.
like
Yes.
electrical endpoint nowadays, or
Yeah.
the real-time detection.
Right, sir.
Like in
So,
those cases, what are the critical issues you are going to address? That was my thing.
The granulation endpoint generally there are two ways basically we see apart from the conventional. So conventional also we are adopting, but that is for our confidence level, how to gain the confidence on the product, and we do have mechanical controls on that. First is impeller amperage. So whatever is the amperage I'm getting during the granulation. So we will be checking the amperage of the impeller as well as chopper during our granulation, amperage of the impeller during dry mixing, and once all the binder is added, so and the end point is reached. So during our trial batch, it shall be monitored that at what stage our endpoint comes. So the endpoint detection depends upon impeller amperage. So to reach the target of particular amperage, as I said previously, I have to optimize a product where during the trial I got an amperage of 30. Okay, so I got the point, and physically also we are verifying that the one we are doing at lab scale also will take the material into the fist, will break the material that lumpy mass into with the help of thumb into two pieces. So also we got the idea from the amperage, and also we got the idea whatever the personal physical experience we are having. So basis upon that, during our next trial, we will be giving the ranges for that amperage, that how much amperage. For example, I got 30. At 30, we got the good granules. So in the next trial, we will be giving the range of 25 to 35. Means 35 plus minus 5 amperage we are giving the range. And we will be challenging that in the subsequent batches. So we can have a endpoint of 28 and we can have a endpoint of 32 also. So during the validation, basis upon the risk assessment and the outcome of the trials, we will be able to assign a particular range. So that range will be able to give the optimum granulation endpoint during the validation. So it will be challenged in the validation.
Okay. So one more thing is that even during the pilot scale also, there should be a provision to measure the amperage, what I can understand.
Yeah, pilot scale also, sir, similar. Nowadays, small scale equipments are available where all these provisions are available. And yeah, those will be displayed into the PLC.
Okay.
Even the printouts nowadays, technology is changed. All the printouts for the complete process, we can get it.
Yeah, Gurucharan, as you know, now we are outdated a bit. So we need to go on updating. Really, to tell you.
That is good, but sir, teachers are never outdated. You are master in the field, and we are still your students and learning. Thank you so much, sir.
Thank you,
thank you a lot, Gurucharan. We're all happy. You're all happy, pa. That's what I feel.
Even I'm more than happy, sir, interacting with you. I'm very much happy.
So
thanks a lot. We'll be in touch.
Sure. And Gursaranjit Singh sir, the last questions and participants, after this we are not in position to take more questions. The last question from Mr. Samir Kochar. Please ask your question, Mr. Samir. This is the last question of session.
Yes, sir. Thank you for your nice presentation. I have just one question, like for process over the proposed range should be established. Whatever range we are proposing, it is necessary to ensure the process robustness through the entire range, and at what scale we should ensure that the proposed range is giving us the same quality of the product. Because sometimes when we are taking trial, we are not taking at the exact scale, at the commercial scale. So we will ensure that whatever ranges we are proposing, that is valid for the scale of the commercial scale.
Okay, fine. So I got your point. So I will just tell you that nowadays scenario is changing in the industry. Okay, and a lot of controls are in place nowadays. I will give you an example of compression machine. If at all, previously once the validation was done, the challenge study was performed. So the challenge study was performed. For example, my compression machine speed is up to 80 RPM. Maximum speed is up to 80 RPM. So I will be doing the compression. So it will be a trial. During the trial, I will be running my compression machine at slow speed. For example, it is 10 RPM. 10 RPM, I will be checking all my in process. I will be doing all those in processes and I will be collecting the sample. Then we will be going ahead for the optimum speed. So optimum speed, for example, if it is 20 or 30, we will be doing all the in processes for optimum speed. And then further, we'll be going ahead for the high speed. It could be 50. So now we are having a range of 10 to 50. In the trial batch or my CB batch, we have done a challenge study of 10 to 50. And my optimum range is where I have compressed as if it is 30. So mean optimum is 30, high is 50. So we are having a complete range. So during the documentation, after the completion of validation, the ranges were given like that my compression speed range is 10 to 50. Okay. What the problem and issues were observed during in the industry? For example, an operator is doing a operation in the night and he feels that my range is 10 to 50, but he is not having any driving or written document where he can get the information that exactly at what speed he has to run the machine. He has to run the machine at 20, 10, 15, 30, 35, 45, 50, because the range is very much wide. So due to that, there were lot of fluctuation. Some operator will try to, अरे यार चलेगा कोई बात नहीं 20 पे भी चला लो. Few will feel अरे maximum चला लो, shift में maximum outcome देना है. But that range is validated. There were certain kind of issues. Nowadays, the concept is that normal operating range and proven operating range is given, NOR and POR. So for example, if at all we are giving a proven operating range, so my proven operating range is 10 to 15 or 50 RPM, and my normal operating range. So normal operating range will be at the range at which the operator will be running the machine continuously. So that will be 30. On 30, we will further give him a range that is small range. For example, 30 plus minus 3 RPM. So now he is having a range that he has to run the machine at 27 to 33. That is his
normal operating
range. And if at all it is required to go further, he can go further. So this is how the ranges are established and defined in the document.
Right, sir. This is for compression, sir. Same can be done for granulation, because granulation we are not varying parameter by much, like say amount of water quantity or kneading time. We generally carried out validation at some fixed parameter or minor variation. So at that time we are not able to get the range which we are proposing for the commercial batches. So there is the issue. How we can resolve that?
That could be controlled. For example, if it is granulation, so granulation is if at all your granulation is amperage based, and whether your granulation is time based. So these are the two basic criteria to control the things. If it is amperage based, so once the data for the amperage is generated, generally whatever the data is generated during the validation, a remark is put into the BMR that these parameters shall be finalized, or amperage shall be finalized after the data generation of the 10 commercial batches. Means after validation, you are having further more batches or which will be run on this range. Till then, these ranges shall be provided to the operator to run the machine. Once the 10 batches data is obtained, so now sufficient data is generated. So now you will be confident that I am having this, I have assigned this particular range, and my 13 batches, 3 validation batches plus 10 commercial batches has run successfully within this range. So you are having the confidence level. So according to that, you can define that range. Okay. And if it is time based, we can control through the time, and we can give that the range for that time. For example, this is my binder addition time from 2 minutes to 3 minutes, and this is my kneading time, this is my wet milling time. So accordingly we can define.
Okay, just last question, sir. The scale of the batches, it should be same as the commercial scale, because every time it is not possible to execute batches at the same scale. So the whatever data we need to generate, it should be at the same scale.
No, it is not mandatory. For example, if at all it is a new product and it is going for a registration to, for example, it is going for emerging market registration, the API is very much costly. So I'm telling my personal experience. So I was in it's ARPO lab, where we have worked on oncology molecules. The oncology molecule API itself is very much costly. One batch cost is near about 20 lakh or 30 lakh, if at all small scale batch we are taking. And if it is pilot scale batch, the cost of the batch will be 80 lakh or 70 lakh, or near about 1 crore. So it is very difficult to take the commercial batch at that, to take the batches of the commercial scale. So what shall be done? 1/10 of the commercial batch shall be taken. Okay. So as per the guideline, 1/10 of the commercial batch or of the 1 lakh tablets, whatever is higher shall be taken. If at all you are going to exhibit a batch of 1 lakh tablets at commercial batch, so you can take 10,000 tablet batch also at pilot scale, and you can submit the data to agency.
Thank you, sir. Thank you for your answers.
Thank you so much.
Thank you, Samir, for your questions and waiting for so long time. So this was the last question from the session. Now we are not in position to take any more questions. And now I would like to say thanks on behalf of whole Kelly College of Pharmacy, the Alumni Association, the teachers, the friends, those who helped me to arrange this webinar series. And friends, I would like to announce to all participants that we are continuously conducting this webinar just to attach or to make a link between industry and academy. So many things we study and which we, when we go to work, we find it uncomfortable while working. So to help webinars, and the presentation by Mr. Gurusaranjit Singh was one from that series. So we'll keep doing like that, and we'll keep you informing, we'll keep you updating for this webinar. So till that, take care, stay safe, and protect yourself as well as your family from this deadly COVID-19, and be happy, my dear friends. So thank you all, everyone. Thank you, Gurusaranjit, for your
Thank you.
wonderful presentation. And
Thank
it
you,
was
sir.
really like I admire your patience. 1 hour 45 minutes. The schedule was for I think 1 hour only. So I'm sorry, question was more. I cannot say no to participants. And Gurusaranjit ji, it also shows that your experience, how much experience you have gained with time in this particular field. And hope it was, I said in the beginning, it was more like a class, and hope the students, the participants has enjoyed and enriched their knowledge skill. So wish you all the best for your future, Mr. Gurusaranjit Singh ji, from Kelly Alumni Association, from your teachers, from your friends, from the whole Kelly College of Pharmacy team. And we wish that you will keep serving the pharma sector, you will keep serving the society with the same zeal, with same enthusiasm, and you will always listen a call from your alumni association. With this note, thank you very much. Stay safe, sir, and we wish you a happy life forever. Thank you,
Thank
sir.
you. Thanks
Thank
all
you,
for
sir.
being a part of this webinar. And I am thankful to Dr. Raman Dong, Principal and Chief Coordinator, Dr. Sujit, Mr. Yuvraj, and Ms. Rekha, and all the other faculty members of KLE for giving me this opportunity to have this session on technical interaction with all of you. And the topic was, as I said, it was a vast topic, and a number of sessions are required for that, and it is not possible to summarize in 1 hour. But still, from students' perspective, I have tried to sum up the things, and that will help them to understand about the topic and some picture about the this topic of scale up and process development. So once of all, thanks to all of you.
Thank you, sir. Thank you. Thank you, sir. Welcome.
Thank you.
Okay, bye, everyone. Take care. Stay safe. Bye bye. Thank you. Yeah, I conclude this session.